is meant by virology? Virology is a branch of science that deals with viruses and viral diseases. In another word, virology is the study of viruses and virus-like agents, including their taxonomy, disease-producing properties, cultivation, and genetics. There are several unique divisions in virology, such as HIV, or as known as human immunodeficiency virus, rabies, influenza, poxviruses, and lastly, hepatitis. Let's find out more about this unique division and its possible test running.
The first unique division of virology is HIV, or as known as human immunodeficiency virus.
HIV or human immunodeficiency virus is a virus that attacks cells that help the body fight infection, making a person more vulnerable to other infections and diseases. It can spread by contact with certain bodily fluids of a person with HIV, most commonly during unprotected sex, sex without a condom, or through sharing injection drug equipment. HIV is most commonly diagnosed by testing your blood or saliva for antibodies to the virus. Unfortunately, it takes time for your body to develop these antibodies. A quicker test checks for HIV antigen, a protein produced by the virus immediately after infection. It is called immunoassay or ELISA test. They are generally very accurate.
Second unit division for virology is rabies.
Rabies is a deadly virus spread to people from the saliva of infected animals. The rabies virus is usually transmitted through a bite. Animals most likely to transmit rabies in the United States of America include bats, foxes, raccoons, and skunks. Meanwhile, in Africa and Southeast Asia, stray dogs are the most likely to spread rabies to people. There are two types of rabies diagnostic testing, which are rabies diagnostic testing in animals and rabies diagnostic testing in humans. For animals, rabies diagnostic testing is direct fluorescent antibody test, which is the gold standard for rabies diagnosis in animals. Animals that are behaving abnormally or show signs consistent with rabies should be tested. This test is performed post-mortem. For humans, their saliva, tears, and cerebral spinal fluid samples are used for virus isolation in tissue culture. Serum and CSF are immediately tested for antibodies to rabies virus in unvaccinated patients.
And the third one is influenza.
Influenza, which known as the flu, a highly contagious viral infection of the respiratory passage causing fever, severe aching, catarrh, and often occurring in epidemic. There are four types of influenza, which is influenza A, influenza B, influenza C, and influenza D. Laboratory diagnosis. First, office-based rapid viral test. This test preferred respiratory sample such as nasal swab or nasal wash, and the sample should be collected within 3 or 4 days of illness. This sample provide result in only 15 minutes later. Second of all, viral culture. This test will run during outbreak of respiratory illness when influenza is suspected. The collection of some respiratory samples for viral culture is essential for determining the influenza A virus subtype and influenza A and B virus strain.
The unit division is poxviruses.
Poxviruses. What is poxviruses? Poxviruses is any of a family of brick-shaped or ovoid double-stranded DNA viruses that have a surface lipoprotein membrane covered with tubular or globular structures.
and that include the Yersinia virus and the causative agents of cowpox, myxomatosis of rabbits, and smallpox. The diagnosis of smallpox. The Center for Disease Control and Prevention can do definitive testing using a tissue sample taken from one of the lesions on the skin of the infected person. Actually, no cure for smallpox exists. In the event of an infection, treatment would focus on relieving symptoms and keeping the person from becoming dehydrated. Antibiotics might be prescribed if the person also develops a bacterial infection in the lungs or on the skin.
Last unit division of virology is hepatitis.
Hepatitis is the inflammation of the liver. The condition can be self-limiting or can progress to fibrosis, cirrhosis, or liver cancer. The causes of hepatitis are: first, autoimmune diseases; second, toxic substances such as alcohol; third, infections. 5 main hepatitis viruses are A, B, C, D, and E. Diagnosis of hepatitis. First is liver function test. For liver function test, a sample of your blood will be taken and analyzed. To get the most accurate results, you might have to fast for 10 to 12 hours before the test. Liver biopsy. In some cases, your doctor may need to examine a liver tissue sample, especially with advanced disease. The sample is taken in a process called a liver biopsy. You will either be sedated or given a local anesthetic, and a tiny sample of liver tissue will be removed with a long needle through a small incision on your right side.
In the nutshell, there are several unit divisions in virology, such as HIV, rabies, influenza, pox viruses, and hepatitis. The possible tests running for HIV are blood or saliva sample test and ELISA test. In addition, for rabies, direct fluorescent antibody test for animals and virus isolation in tissue cultured for humans. As for influenza, office-based rapid viral test and viral culture. Furthermore, for pox viruses, tissue sample test, but there is no specific cure for smallpox. Last but not least, for hepatitis, liver function test and liver biopsy.